So to test this now, I perturb the antigen signal just by culturing the cells without an antigen stimulus. And here I measure T-Helfer expression after 5 hours of stimulation with interferon gamma. And you see that it's strongly induced, and in the absence of antigen, the levels are decreased. So this was expected because here antigen is a positive regulator of T-Helfer. But now when I measured in the same experiment the expression level of the IL-12 receptor expression, I saw the opposite effect. That in the absence of antigen, the levels are much higher, suggesting that the antigen signal actually has a repressive effect on the expression of the IL-12 receptor. So therefore, I included this additional new regulatory link in the model, and now it could explain quite well its biphasic expression kinetics. And in addition, I also measured a larger data set. Where you see here there's T-Helfer, IL-12 receptor, and interferon gamma, in these different cultural conditions that I showed already. And you see that the orange line, which represents the completed model, can actually simulate quite well the behavior, while the original literature-based model as a dotted line did a pretty bad job. So now, if we look at the structure of this completed network, we see that there is now a second positive feedback loop that controls T-Helfer expression, which is mediated by the IL-12 receptor and IL-12. And the antigen signal now acts as a switch between these two T-Helfer induction pathways, because the antigen activates interferon gamma-dependent T-Helfer expression, but it represses IL-12-dependent expression. So therefore, this model would predict that if you vary the duration of the antigen signal, that the system should switch between these two pathways at a different point in time. And this is illustrated here with the simulation first. So in red, I simulated that the antigen signal was only present for 1 day, and you see that here the second phase of T-Helfer is induced earlier together with the IL-12 receptor. And then to test this prediction experimentally, I blocked antigen signaling after 1 day with a chemical inhibitor, and you see essentially the same effect of the second phase of T-Helfer. It starts up earlier together with the IL-12 receptor. So I think from the data I presented, really suggests that this completed network structure now captures the main regulatory mechanisms that control these 3 genes during Th1 differentiation. So therefore, in the last slide, I want to come to the second question of how this network now controls differentiation. So this is what I showed, that I measured these expression kinetics during 6 days of differentiation, but then for each experiment, I stimulated the cells again at the end of differentiation after 6 days and measured how many of these cells would produce interferon gamma. So this is a way to quantify the differentiation essentially. And then I looked how the expression levels during the differentiation process were connected to the outcome of differentiation. So what's plotted here is the mRNA expression after 1 day of differentiation in the early phase, and on the x-axis is the memory for interferon after 6 days, and each of these symbols represents one experiment. You see that there's essentially no correlation, so it's not important whether T-Helfer is expressed in the early phase for the outcome of differentiation. But if I do the same analysis now at day 4, so in the second phase, you see a very strong correlation now, suggesting that T-Helfer, specifically in the late phase of priming, plays a functional role for driving differentiation. So taking together all the results I've shown to you, we can now answer this initial question of why IL-12 is so important. It's because IL-12, but not interferon gamma, can induce T-Helfer expression specifically in the late phase when T-Helfer is required for differentiation. So, in summary, this approach of combining modeling with the quantitative measurements really allowed me first to find 2 new regulatory links, and then to answer really a long-standing question on the molecular immunology about the mechanism of IL-12 dependent Th1 differentiation. So with this I want to end, and I'm happy to take all your questions. Thank you.
Thanks a lot for the very nice presentation. Are there questions from the audience?
in the medium. Also, there is not really a source of IL-2, but of IL-4, but it can in principle be produced by the T-Helfer-Cell. That's why I just try to have a clean system which blocking all the alternative pathways.
I have a question. How can you be sure that only these 3 players are involved? Maybe there are very complicated detours and regulation via some other cytokines.
Yeah, no, this is of course a very important question. So I'm also not saying that there's only these 3 players involved. So first of all, the regulatory links in my network, they don't necessarily correspond to a direct regulation. So they can definitely other places that mediate these interactions. So I could not exclude. Also for some things I've looked at this stuff. And second, if there are, so I think they are central regulatory genes in this process that are already able to explain a lot of the things that are going on. But then, when I started this project, I didn't expect that it would be sufficient to look only at the 3 genes. So I expected that you start with a small number of genes, and then you find that there's something that I cannot explain, and you need to look for another factor that might be responsible. But it turned out that these 3 genes were essentially sufficient to explain quite a complex set of data, suggesting that you have, so how I see it is more that you have sort of a hierarchical regulation, so these 3 central players, and then you have probably other layers of regulation that mediate their effects and so on. But so for this, I think there's no other cytokines that's essential, I would say, but they cannot completely.
Okay, I would like to thank the speakers again.